Good morning, students. So today I will be discussing on the topic that is your Streptococcus species. So as we have already known that Streptococcus species is one of the very important organism of clinical significance because Streptococcus can cause numerous arrays of diseases. Now first and foremost thing we need to know that in what risk group does this Streptococcus fall. So this Streptococcus fall on this risk group 2. This is a very important fact one microbiologist or anyone who is going to a microbiology lab should know. That's risk group means what? So Streptococcus falls under risk group 2, which means that Streptococcus has the ability to cause disease, but there are many medications or treatments or prevention which can stop this disease, and hence the mortality rate is low. Now, if we move on to the general characteristics of Streptococcus, it involves that it is grams positive. Now, if I talk of gram positive, what is gram positive? Gram positive is based on the cell wall characteristics of an organism. That means this Gram's character, that is Gram's character means when you do a Gram staining, which has been discovered by Hans Christian Gram, what type of color it will provide or it will be showing after the staining procedure. So depending upon the cell wall composition, gram positive shows a purple color. So what is the basic principle behind it, I will be discussing in my later videos, but for the time being you understand that it is grams positive according to the gram staining process, and it is spherical, which is a very important point, that it is spherical. What is the shape of it? And it is having a default shape, that is it is oval cocci. Now this diagram is very important, as you can see that you get to see that this oval cocci are in chains. So when you see any cocci in chain, then it is from Streptococcus genera. If we go back into the Griffith experiment has dealt with transformation, where he has taken this Diplococcus pneumoniae. Diplococcus means two coccus one after the other in pairs, but when they start to multiply in this plane, you see that the plane is in this way, it is multiplying, it will ultimately result in a chain, and that's why we call it Streptococcus, that is cocci in chain. Now there is another important classification of Streptococcus depending upon hemolysis. Now students, you may be thinking that what is hemolysis and why it is important. This is because there are certain group of organisms which are called fastidious group of organism. Like for example, some child will not eat food if some important platter is not given in his or her plate. So that is called fastidious. So this organism will only grow when some RBC is supplemented in their medium, and they multiply in the RBC, resulting in rupturing of the RBC, lysing of the RBC, and resulting in hemolysis. So depending upon the hemolysis, which can be partial hemolysis, right, then there is a complete hemolysis. So you have alpha hemolysis, you have beta hemolysis, and last but not the least, gamma hemolysis. So when it is alpha, it is partial. When it is beta, it is complete. When it is gamma, it is no hemolysis. And depending upon it, you will have this classification. I will tell you the basis that what is A, B, C, D and G, but for the time being you understand that those who undergo this beta hemolysis, they are A, B, C, D and G. I will tell you the basis of classification. An example is Streptococcus pyogenes. So my question to all of you, you will be writing down in the comment box that what is the clinical significance of Streptococcus pyogenes, why it is called pyogenes. So these are my two questions in this video. You can write down in the comment box of the video. So next is your alpha hemolysis, which is your partial hemolysis, and I think that all of you are acquainted with this pathogen, that is Streptococcus pneumoniae, that is the causative agent of pneumonia. It's a very common disease, and Streptococcus viridans, right? So beta hemolysis, gamma hemolysis. Moving on to alpha hemolysis. Now when I am talking of alpha hemolysis is associated with Streptococcus faecalis. Now gamma hemolysis is actually no hemolysis. So if it is Streptococcus faecalis, then you will have no hemolysis if you grow them in blood agar. So in a nutshell, I can tell you that if I grow the pathogen in a blood agar, that means an agar which is basically supplemented with the sheep blood, and I try to streak the microorganism, what is happening? This microorganism will undergo lysis all around. The RBC will be lysed. Sometimes there will be partial lysis, sometimes there will be complete lysis. Accordingly, you classify them as alpha, partial, beta, complete, gamma, no hemolysis. And then we can give an example of beta hemolysis having Streptococcus. It doesn't mean that only Streptococcus is causing this hemolysis. Staphylococcus do cause hemolysis. So for Streptococcus pyogenes, it is beta hemolysis. For Streptococcus pneumoniae, it is alpha hemolysis. And for Streptococcus faecalis, it is gamma hemolysis. Moving on to some other important characteristics of this Streptococcus species. The first, gram positive, which I have already discussed. Next is your non-spore. Now non-spore means what? They do not form any kind of spore. You know that there are two kinds of spores, that is your endospore and exospore. That means a spore which is present inside the cell and outside the cell. So they do not form any spore. That means what? They are unable to survive the stress. Stress means when there is dehydration, when there is desiccation, right? If they are subjected to any kind of chemicals, so they will not be able to survive the stress, as because they are non-spore forming. Next is non-motile. This is also one important fact. We can do a lot of experiments about motility of a bacteria, right? And usually they have flagella who are motile, who undergoes motility, but this organism do not bear such motile appendages, and hence they are non-motile. Another important point, this is basically your capsules. So they even form capsules. That is a very, very important point, that they can form capsules. Capsules, if I highlight upon it, it is basically a virulence factor. It is basically a virulence factor. So this is a very important point, that is that they form capsules, right? That means this is one of the virulence factor. Again, I go back to the Griffith experiment, that is on Diplococcus pneumoniae. If you have remembered this Griffith experiment, which was usually taught in class 11 and 12, that scientists have worked with two type of strain, R strain and smooth strain, and he talked that R strain or rough strain was a virulent strain, whereas smooth strain is your virulent strain. That means a virulent, that means it cannot cause any disease. And why it was not causing any disease? Because it do not have a capsule. It is having a rough colony. From the rough colony, it was called R. So immune system can easily understand that particular microorganism and eliminate from the system, and thus it doesn't cause pneumonia. But when smooth strain was injected into the mice died. And smooth strain is because they form glistening colony, as because they were forming an exopolysaccharide, which is your capsule. Though I try to tell you one thing while I discuss all these things, that students always, exopolysaccharides is not a component of capsule. There are many organisms, like for example Bacillus anthracis who do not have an exopolysaccharide as a capsule. So my another question from this video is that what type of capsule do it have? Because Bacillus anthracis is a very deadliest pathogen. It also form a capsule, but it is not an exopolysaccharide, right? So what type of capsule does it form? So again we move back to this smooth strain. So smooth strain is because they are forming capsule, they are hiding from the immune system, and that is why they can survive and cause the disease. So that is the importance of capsule which I have discussed. Next is its fastidious. I told you from the very beginning, depending upon its fastidious nature, we have classified it as alpha, beta and gamma hemolysis, because we have to supplement the media with a red blood cell. Usually sheep blood is being used for their growth, right? So even if you are doing a sputum analysis of a patient suffering from pneumonia or upper respiratory tract or lower respiratory tract infection, you have to use this blood agar for confirmation of the presence of Streptococcus. And moreover, the colonies are non-pigmented. Why it is very important? Why I am trying to focus on it? Because in the last video, where I have done on Staphylococcus are usually pigmented. They are also risk group 2 pathogen, but they are basically pigmented pathogen. That means they have in Staphylococcus you are classified into 3 colors like golden yellow, lemon yellow and white colored colonies. So when you go into the colony characteristics for Streptococcus, you do not find such pigment. So it is non-pigmented colonies. So I basically sum up the today's video by saying that Streptococcus this is just the first part of the video, because a lot of information need to be given for Streptococcus. But for Streptococcus species, it is a risk group 2 pathogen, and moreover it is a cocci in chain with Gram's character as gram positive, followed by it can show hemolysis, right? Alpha beta, partial and complete hemolysis. Examples are given like S. pyogenes and S. pneumoniae and S. viridans for alpha hemolysis. Even gamma hemolysis, that is no hemolysis. So S. fecalis, right? And further, if we go into the other characteristics of Streptococcus, which comprises of non-spore, so they cannot survive the stress, non-motile, they do not have any motile appendages, they have capsules, which is a virulence factor which can result in the disease, and that's why elimination of this pathogen is a bit difficult, because immune system takes a lot of time to eliminate and clear out this pathogen from the system. And it is fastidious. Without its required cell, that is RBC, it is very difficult to grow. That's why it is grown or supplemented. Agar is supplemented or nutrient is supplemented with sheep blood or RBC. And moreover, if you see the colony under the microscope, under your in a plate, you will get to see it's a non-pigmented colony. So with this, I end my first part of the video. Next, I will move on to other video next day, which comprises of how it is transmitted, what are the etiology and the pathogenicity, pathophysiology of various diseases associated with Streptococcus and its diagnosis and treatment. Thank you.